Hi, thanks for opening this video. Today we are going to study about transport across the cell membrane. As we saw before, we've studied about the cell, we know about the cell membrane. So the transport which happens between the cell and its environment, or how the cell interacts with the environment, it all happens via the cell membrane surface. So let's without further ado, let's dive into the topic. So the internal environment of a cell is maintained differently from that of its external environment by a thin surface membrane called a cell or plasma membrane. Like the cover of the cell, we can call it a cell membrane or a plasma membrane. The plasma membrane is a lipid bilayer. It is a lipid bilayer. It means it has two layers of lipid with a phosphate heads and fatty acids tails. Those lipid layers, they have the heads which are phosphate and their tails are the fatty acids. So what are the use, or what is the use of the plasma membrane? The plasma membrane governs the entry and exit of molecules and ions. Like the use of a cell membrane is to govern the entry and the exit of molecules and ions in or outside the cell. This property of a plasma membrane that regulates the exchange occurring between the cell and its medium is referred to as cell permeability. Cell permeability is like a property of a cell membrane which allows it to regulate what enters the cell. A cell membrane therefore determines which substance can enter the cell, comprising those which is the important vector, and also regulates the outflow of substances containing excretory waste. Like here we need to see or to know about which which kind of substances that the cell membrane allow to enter the cell, and which kind of substances that the cell allows to outflow outside the cell. So the first the types of substances that the cell allows in the cell, that the cell membrane allows in the cell, is comprising those substances comprise those which may be important to the vital activities of the cell, like all things that the cell uses to for each vital activities, for respiration, respiration, for feeding, and many other things. It also regulates the outflow of substances. Those substances are the excretory waste and water. Like the kind of substances which goes out of the cell are the excretory wastes and water. This feature of the membrane this feature of the membrane not only maintains difference in composition of intercellular and extracellular fluid, but also some substances were impermeable to others. It's called selective permeability. If we say the cell membrane is selective permeable, it means it allows certain things to enter and refuses for others to not enter. So gradients across the plasma membrane. Let's see what we call a concentration gradient. A concentration gradient refers to the difference in the concentration of a substance from one region to another across a plasma membrane. In such a case, the solute will have a tendency to move from a region of high to a region of low concentration. If we say concentration gradient, simply is the difference between concentration on the sides of a surface. Like one side can have too much concentration of a certain thing, and another side of the surface can have too low concentration of something. So they are saying that the substance move from a region of high to a region of low concentration, a region of high to a region of low concentration. Due to the distribution of positive and negatively charged ions, the inner surface of a plasma membrane is more negatively charged than the other. Inside the cell is more negatively charged more than it is outside the cell. So the difference in the electrical charge between two regions create what we call electrical potential. We call it electrical potential because the the number of charges are not equal on either side. Since this is established across a plasma membrane, it is termed as a membrane potential. We call it a membrane potential because it happens around the plasma membrane. So let's go into movement of substances across plasma membrane, like how do we classify the movement of substances across the plasma membrane, like which kind of substance moves, or in how way or in which way does a substance move, and how do we name it. As mentioned before, plasma membrane is selectively permeable. It lists all the processes involved in the movement of substances across cells. Here we have the example. You actually have to memorize these ones. Bino mukwiye kubyiga nyine mukabifata mu mutwe. We have the first way cyangwa se the transport across the cell, which we call passive transport. The passive transport has three main types. We have simple diffusion, osmosis, facilitated diffusion. And we also have primary active transport, the primary active transport, and also a secondary active transport. All these three, passive transport, primary active transport, secondary active transport, we call they transport small molecules. We also have endocytosis, which has two types: the phagocytosis, pinocytosis, and also we have exocytosis, which have constitutive exocytosis, regulated exocytosis. All these transport large molecules. Then the transport mechanisms can be broadly classified into two types. Actually, these are the transport mechanisms. We put them into two main types. We call them passive transport. A passive transport, it involves the movement of molecules along the electrochemical gradient without the use of ATP. That's what we have to put in mind. When there is movement of substances across the cell membrane without any need of using energy or ATP, we call it passive transport. Or we just like say that it is a downhill transport, like something when something ikintu kiriho kiramanuka nk'ahantu ku musozi nta mbaraga gikoresha, kiramanuka gutya nta mbaraga. Rero ibintu tuvuga yuko habaho passive transport iyo substances zavuye cyangwa se zikinjira muri cell hatabayeho gukenera gukoresha imbaraga cyangwa se energy. So icyo gihe rero harimo za diffusion ndetse na osmosis. Hanyuma iki twita active transport, it drives the molecules against their electrochemical gradient by hydrolysis of ATP. Actually here iyo hari kubaho transport bigakenera yuko turi buze gukoresha energy, ibyo ngibyo tubyita active transport. Impamvu byibutwa active transport nuko actually substance ibiri kujya ahantu naturally itakakwiye kujya. Nuvuga nko mu busanzwe twabonye yuko substance iva ahantu hari ifite high concentration ikajya ahantu ifite low concentration. Rero iyo turi guforcinga ko ikintu kiva ahantu hari low concentration kijya ahantu hari high concentration, icyo gihe bivuga yuko itari kugendera kuri electrochemical gradient. Icyo gihe rero bigakenera yuko dukoresha energy, cyangwa se kugira ngo energy iboneke hasabaho ko habaho hydrolysis of ATP. That's why we call it uphill transport. Uphill transport nk'uko umuntu azamuka umusozi. Twagiye rero muri passive transport, twabonye ko dufitemo diffusion ndetse na osmosis. So passive transport, it is the simplest mechanism in which a molecule dissolves in the phospholipid bilayer. As we said, the cell membrane is composed with the phospholipid bilayer, and the substance diffuses across it and then dissolves in the aqueous solution presented on the other side of the cell membrane. It neither requires ATP or any protein. Actually, we don't need any protein or ATP usage while we are transporting something across the cell membrane, and the direction of movement is determined by the concentration gradient. As we said earlier, the molecules move from a region of higher concentration to a region of lower concentration. Uvuga ngo n'ubundi ikintu iyo kiri ahantu ari cyinshi ahandi ari gikeya kivaho kiri cyinshi kijya ahantu kiri gikeya. So therefore any molecule that is soluble in phospholipid bilayer is capable of crossing the plasma membrane. This is the reason why only small relatively hydrophobic water repelling molecules, example benzene, gases, oxygen and carbon dioxide, and even small polar and charged molecules diffuse easily across the plasma membrane, while the other larger molecules are restricted. So diffusion also is also regarded as the random mixing of particles in which particles continue to move down their concentration gradient until an equilibrium is reached and particles are evenly distributed. Ni ryari diffusion ihagarara? Diffusion ihagarara igihe particles, ni ukuvuga ngo dushobora kuba dufite inyuma ya cell, dushobora kuba dufite wenda nk'urugero dushobora kuba dufite substance called A iri nyinshi kurusha uko irimo mo imbere. Hazabaho transport by diffusion mpaka igihe tugezaho concentration on both sides zingana, icyo gihe nibwo diffusion igira gute ihagarara. So what are the factors which affect the rate of diffusion? Ikintu cya mbere, the greater the concentration difference between the two sides of the membrane, the faster the rate of diffusion. If there is too much difference between the concentrations on two sides, that time the diffusion goes very quickly at a higher rate, and as the temperature increases, the rate of diffusion increases. If the temperature increases, like the temperature ibaye nyinshi, diffusion nayo igira gute irakorwa byihuse. Smaller molecules have faster rate of diffusion, while the ones with larger mass diffuse slowly. Nuvuga ngo ama molecules ari matoya adiffusinga byihuse kurusha molecules ziri nini. The larger the surface area of a membrane available for diffusion, the higher the rate of diffusion. The greater distance across which diffusion is to occur, the longer it takes for molecules to pass through. Ni ukuvuga ngo iyo surface iri nini kugira ngo diffusion nyine ibeho yisanzuye, na rate of diffusion nayo iba ndende cyane nini. The greater the distance across the diffusion is to occur, like iyo harimo distance nini hagati y'ama membrane, icyo gihe diffusion iragira gute iratinda, imara umwanya munini. So what are the significance or the importance of diffusion? Like diffusion ikora akahe kazi cyangwa se ifite uwuhe mumaro? In the human body, nutrients in the form of ions and small molecules are observed from the food by the surrounding blood cells in the vessels by way of diffusion. Ni ukuvuga ngo mu mubiri w'umuntu ibiribwa intungamubiri zirimo imyunyungugu ndetse n'amamolecules matoya aba absorbed muri blood vessels cyangwa se muri blood cells mu mitsi y'umuntu hakoreshejwe uburyo bwa diffusion. Ni ukuvuga ngo simply intungamubiri zigera muri blood cells zawe gute? Zigeramo hakoreshejwe inzira ya diffusion. And in lungs, carbon dioxide diffuses out of blood in alveolar sacs, whereas oxygen present in high concentration in the inhaled air diffuses in the cells in the blood vessels with low oxygen concentration. Ni ukuvuga ngo muri lungs cyangwa se mu bihaha, carbon dioxide ijya mu isohoka mu maraso y'umuntu ijya mu tufuka tubika umwuka hakoreshejwe diffusion. Same thing happens kuri oxygen. Cutaneous respiration through skin is the most common mode of respiration in lower non-chordates, wherein gases directly diffuse through the air in the surface epithelium of the organisms. Ni ukuvuga ngo hari animals zihumekera mu ruhu, the animals rero zihumekera mu ruhu kugira ngo zihumekere mu ruhu, umwuka usohokera mu ruhu by diffusion. So the eyes lack great number of blood vessels which carry oxygen, therefore needs an extra supply of oxygen. The atmosphere provides this extra needed oxygen, which is taken up by the eye through direct diffusion of oxygen in the cornea, the hard outer covering eye also. Nuvango mu busanzwe mu jisho harimo imitsi mikeya izamura amaraso ku buryo nyine hashobora kuboneka oxygen ihagije. Rero icyo gihe oxygen ituruka mu mitsi imwe imbere mu maso ntabwo iba ihagije, niyo mpamvu dukenera oxygen iturutse hanze. Oxygen iturutse hanze rero amaso ayabsorbinga by diffusion nyine mu kirere, akinjira nyine mu ruhu hakoreshejwe diffusion. Ibyo bintu bitabaye amaso yakumagara. For medicines taken orally as pills, the medicine must somehow find its way into the bloodstream. No kuvuga ngo iyo unyweye umuti w'ikinini, then kugira ngo uwo muti ubashe kugera mu mubiri biba bisabwa yuko nyine winjira mu maraso. Rero uburyo umuti winjira mu maraso nabyo ni process of diffusion ibaho. Gases exchange during the process of respiration and photosynthesis takes place with the help of diffusion. Gases exchange muri plants ndetse n'uburyo bwa photosynthesis byose bibaho hakoreshejwe the diffusion as a way of transport. So transpiration or loss of water from the aerial parts of the plants involves the process of diffusion. Uburyo ibice byo hejuru by'igiti bigenda bibura amazi nabyo bibaho hakoreshejwe uburyo bwa diffusion. Diffusion is involved in passive uptake of mineral salts. Other of the flowers attract the pollinating animals, spreads the air by diffusion. Umwuka cyangwa se impumuro nziza ituruka mu ndabo, iyo mpumuro nziza igera mu kirere hakoreshejwe uburyo bwa diffusion. So diffusion plays an important role in imbibition and osmosis. Diffusion nayo irafasha mu kintu cyitwa osmosis and imbibition. Imbibition ni iki? Imbibition is a special type of diffusion involving the absorption of water molecules by solids such as absorption of water by wood or seeds. Nuvuga ngo imbibition ni uburyo ikintu nyine kiri solid cyabsorbing amazi cyangwa se kinyunyuza amazi. Tuvuze nk'urubaho ni gute rufata amazi akarugeramo, ese iyo uteye urubuto ni gute urubuto rukurura amazi mpaka rwinjiyemo. Ibyo nibyo twita imbibition. Then twagiye rero ku cyo twita osmosis. In osmosis, the movement of water solvent occurs due to the difference of chemical potential by water potential in case of. So I actually have to say something here. Twize chemistry, tuzi solvent tukamenya na solute. Muri diffusion iyo hari kubaho transport, habaho transport of the solute. Hanyuma muri osmosis iyo hari kubaho transport, habaho transport of solvent cyangwa se water. Hanyuma mechanism y'uburyo bigenda byo ni bimwe. Nuvuga ngo byo ni bimwe rwose. Mechanism n'ubundi biva ahantu biri byinshi bijya ahantu biri bikeya. It means muri osmosis ni transport y'amazi. Amazi ajya ava ahantu ari menshi akajya ahantu ari make. Nampamvu rero bazumva osmosis buri gihe iba associated with water, ariko baba bashatse kuvuga solvent nuko nyine water ariyo universal solvent. So osmosis rero is the movement of water or solvent molecules from the region of their higher chemical potential cyangwa se free energy to the region of their lower chemical potential across the semi-permeable membrane. I've explained about that. Hanyuma rero, if the medium is hypotonic with respect to the cell, it has solute concentration lower than the cell in interior. Nuvuga ngo tukeneye kumenya icyo twita hypotonic and hypertonic solutions. Nuvuga ngo iyo substance cyangwa se solution izaba hypotonic with reference cyangwa se with respect cyangwa se tuyigereranije n'indi solution iri muri cell. Nuvuga ngo rero solution izaba hypotonic with respect to the cell izaba ifite solute concentration iri hasi kuri cell interior. Icyo gihe rero water will tend to move to the cell. Icyo gihe amazi iyo tuvuze ngo ubwo iri hypotonic tuvuze ko ifite amazi menshi, noneho ubwo ifite amazi menshi ubwo mo imbere muri cell ho harimo amazi make, icyo gihe rero amazi azaturuka muri ya solution iri hanze ya cell, then yinjiremo imbere muri yinjiremo imbere muri cell itari ifite amazi, tubone cell iri kugenda iswellinga cyangwa se igira gute ibyimba. Icyo gihe rero tuvuga yuko yabaye tagged, tagged. Hanyuma here is the diagram. Cell placed in hypotonic solution. If a cell is placed in hypotonic solution, the cell swells and bursts or leaves. Hanyuma if a cell is placed in hypertonic solution, hypertonic solution ni solution ifite concentration nini kuri cell, ubwo bikavuga yuko ifite amazi make, bivuze ko amazi noneho azaturuka muri cell yinjira muri solution, niyo mpamvu tuzabona cell ihombana cyangwa se ibura amazi. Hanyuma iyo dufite rero isotonic solution ni solution inganya concentration na cell na interior of the cell, icyo gihe there is no movement, bizaguma uko biri kubera yuko amazi kuri side zombi aranganwa. Icyo gihe ndumva icyitwa iyo solution yitwa hypotonic mwayumvishe, hypertonic mwayumvishe ndetse na isotonic. Hypotonic ni solution ifite concentration cyangwa se ifite concentration ya solute nini kuri ntoya, concentration ya solute nini ntoya kuri cell, ubwo ikagira solvent nini. Hanyuma hypertonic iba ifite solvent ntoya ikagira solute nyinshi, hanyuma isotonic biba binganya. Nkuko twabyivuze, osmosis turaba duturuka ahantu amazi ari menshi tujya ahantu amazi ari make. Hanyuma hari icyo twita reverse osmosis, ariyo iri important cyane nanone. It is used as a process for water purification. Reverse osmosis bayikoresha mu buryo bwo gusukura amazi. It uses pressure in excess of osmotic pressure that forces water to pass through the semi-permeable membrane. As a result, all the solute are retained on one side, whereas pure water accumulates on the other side. Hanyuma tubijyamo cyane, is something that you can read. Osmosis muri animal cells, osmosis muri plant cells, it's simply the same thing. You can read about this. Plants, how they behave in certain conditions, we've studied about that. Then let's go to the active transport. Active transport, as we said, is the movement of ions or molecules from a region of lower concentration. Vango muri active transport hono n'ibintu biri guturuka kuri lower concentration bijya kuri higher concentration across the plasma membrane, cyangwa se tubyita uphill transport. For this, the energy is provided either by another coupled reaction or by direct hydrolysis of ATP. Kugira ngo ikintu kibe cyava kuri lower concentration kijya kuri higher concentration, urumva birasaba energy. Rero icyo gihe uburyo bwo kubona energy hashobora kuba hari ama reaction yagiye abaho, cyangwa se hagasabwaho ko habaho hydrolysis of ATP. ATP murabizi yuko ni energy molecule. So primary rero active transport involves direct hydrolysis of ATP. Vango habaho direct hydrolysis of ATP, icyo gihe tuvuga ko turi muri primary active transport. Example includes ion pumps, for example sodium potassium pumps are responsible for maintaining gradients of ions across the plasma membrane. Hanyuma ikintu cyo kwifata nyine cya mbere nuko habaho direct hydrolysis of ATP muri primary active transport. Then dufite secondary active transport. Active transport driven by ion gradients. Molecules are transported against the concentration gradient, not using energy derived directly from ATP hydrolysis, but coupled with the movement of second molecule in energetically favored direction. No kuvuga ngo hano muri secondary active transport ntabwo twita dukora hydrolysis ya ATP ako kanya, ahubwo the movement of one substance itera movement y'iyindi, like when substance runaka imuvinze bituma n'indi substance nayo imuvinga muri opposite direction. Icyo gihe rero tukavuga yuko the movement of one substance is forcing the movement of another. Nk'urugero, from higher concentration to lower concentration, for example glucose is transported coupled. Iyo tuvuze coupled nyine like biragendana, or coupled transport of sodium ion. Sodium ion gradient is responsible for transport of glucose against concentration gradient from the intestinal lumen of the cell. Nuvuga ngo tugendeye kuri gradient concentration ya sodium, hakabaho transport ya sodium, bituma habaho transport nanone ya glucose. Yeah. Hanyuma when iyo hari kubaho movement when two molecules transport in the same direction tuvuga ko ari sympot. Hanyuma iyo dufite molecule imwe iri kugenda iri kugenda tubyita unipot. Ubwo ni nka diffusion wenda ya glucose n'ibindi bitandukanye. Hanyuma iyo hari kugenda two molecules ariko muri opposite direction we call that antipot. Nk'urugero sodium na calcium buri antipot transport. Another example ni sodium na hydrogen ion nabyo biri antipot. Hanyuma factors affecting the process of active transport. It is known that active transport is carried out with help of pumps. There are two factors which importantly affect the active transport, including the rate of transport by individual active transporters and the number of active transporters present in the membrane, or in another term, the surface. Furthermore, the rate of transport by individual transporter in turn depends upon the nature of transporter, electrochemical gradient, or electrochemical driving force, or either side of the membrane. Uvuga ngo ibintu bya affecting the rate of transport of molecules harimo uburyo binyura muri pumps zazo. Ikindi uburyo nature ya transporter, electrochemical gradient cyangwa se gradient concentration differences between two sides. Hanyuma rero ni uwuhe mumaro wa active transport? Active transport ifasha mu bintu byinshi. The active transport is the intestinal line in the muri ntuzi active transport. Nk'urugero ni gute turabizi ko muri digestion mumara hari uburyo amara nayo yabsorbinga nutrients. Nuvuga ngo rero nutrients especially nka glucose iba absorbed mu mara hakoreshejwe active transport. Hanyuma sodium and potassium gradients established by sodium potassium pump is required for the propagation of electric signals in nerves and muscles. Nuvuga ngo the sodium and potassium pump, iyo sodium na potassium biri kuba transported across muri protein imwe imbere, icyo gihe bifasha mu kugenerating electric signals mu mitsi ndetse no muri muscles z'umuntu. Hanyuma dufite calcium ion are actively transported by calcium pump which is required for muscle concentration. Calcium ion iba transported na calcium pump ari yifasha muri muscle contraction. Hanyuma ion plus ions, hydrogen ions are actively pumped out of the cell lining the stomach, which results in the acidity or gastric fluids which help in the digestion. Nuvuga ngo inafasha mu bintu by'igogora with hydrogen ions. Hanyuma rero twagiye kuri endocytosis. Christian de Duve coined the term endocytosis, which is responsible for the ingestion of large particles such as bacteria, micromolecules, and fluids into the cell in the form of small vesicles. Unlike the above-mentioned processes involved in the transport molecules, endocytosis is only means by the which large molecules or particles can be taken up by the cell, especially in eukaryotes. Nuvuga ngo ikintu cya endocytosis cyashyizweho mu buryo cyangwa se cyasobanuye mu buryo bwo kwerekana uburyo particles nini nk'amabacteri zishobora kwinjira muri seli. Mbere mu bintu twavugaga amaparticles matoya, ama ions like sugars, utuntu nk'utwo dutoya, ariko ibintu binini nk'amabacteri n'ibindi cyangwa se ibyo twita macromolecules ni gute zinjiramo imbere muri cell. Byinjiramo muri process yitwa endocytosis, aho dufitemo phagocytosis izwi cyane nka cell eating. It involves ingestion of bacteria, cold debris or even intact cells. Hanyuma tukagira icyo twita pinocytosis, ari icyo twita cell drinking, aho involvinga uptake of fluids by the cell. Then twagira kuri phagocytosis. This serves as means of food capturing by bacteria and many protozoans, while in eukaryote tugiye kuri side y'ama unicellular organisms zikoresha phagocytosis mu buryo bwo gufeedinga cyangwa se mu buryo bwo kurya. Hanyuma muri eukaryotic cell dukoresha cells zacu zikoresha phagocytosis mu buryo bwo kwirinda cyangwa se bwo kuturinda organisms mbi ziduteza indwara, uburyo bw'ubwirinzi. Nuvango nka zino cells, white blood cells, turabizi yuko arizo zishinzwe defense y'umubiri w'umuntu. Uburyo zikora ibyo bintu nuko white blood cell igenda ikagira gute, igakavaringa cyangwa se ikinjectingamo cyangwa se ikakora ingestion y'amabacteri cyangwa se y'ama foreign body, hanyuma ikadestroyingiramo mwimbere. Hanyuma you can see uburyo phagocytes cyangwa se uburyo phagocytosis igenda igafata bacteria ikinjiramo mwimbere mu mubiri, then ikagira gute, ikayidestroyinga. Hanyuma rero muri pinocytosis ho ni nko kudrinkinga, nuvango it is called fluid endocytosis. Iyo hari kwinjira ibintu biri fluid by'amatembabuzi cyangwa se biri fluid bidakomeye byinjiramo imbere muri cell, tubyita pinocytosis. Hanyuma dufite icyo twita exocytosis. Exocytosis ho simply ni ugusohora ibintu hanze. Nuvango endo ni ukwinjiza, exo ni ugusohora. Muri endo twinjizaga ibintu biri large molecules mwimbere mu mubiri in form of vesicles. Hanyuma muri exo ahubwo ho turasohora ibintu hanze ya cell. Icyo gihe tuzabyita iki? Tuzabyita exo. Hanyuma reka turebe the difference between endocytosis and exocytosis. Muri endocytosis, eukaryotic cells taken up micromolecules from the surrounding by endocytosis that cannot pass through the cell membrane. No kuvuga ngo hakoreshwa ibintu byinjira mu buryo bwa endocytosis ku buryo kubera yuko bitabasha gupasinga muri cell membrane byoroshye. Hanyuma dufite it helps to ingest molecules to the interior of the cell. Nuvango muri endocytosis molecules zinjiramo imbere muri cell, ariko muri exocytosis the cells expel large molecules of particles that cannot pass through the cell membrane. Cell isohora ibintu bitashoboka ko binyura muri cell membrane, ikabisohora hanze mu buryo bwa vesicles. Hanyuma it helps in expelling molecules outside the cell. Iyo ni exocytosis, twavuze ko nyine ari gusohora ibintu hanze. Hanyuma habaho building of vesicles muri endocytosis, ahubwo noneho muri exocytosis hakabaho destruction of vesicles. Hanyuma rero dufite it can be further categorized into the following two. Ifitemo phagocytosis ariyo cell eating, ikagira cells taxing food and what. Hanyuma tukagira pino ariyo cell drinking, aho cell ingestinga ibintu biri liquid material. There is no further organization or categorization muri exocytosis. Hanyuma the material to be internalized is surrounded by plasma membrane which buds off inside the cell to form a vesicle containing the ingested material. Iyo ni ku buryo process igenda. Hanyuma molecules to be transported are surrounded by vesicles which move towards the cell membrane and get attached to it. Molecules are pushed out of the membrane. It leads to the ibyo byo twabivuze. Hanyuma example, uptake of nutrients, food particles, proteins and specific molecules, destruction of phagocytes, pathogen by cell. Ibyo byose ni process ikoresha endocytosis. Hanyuma tukagira secretion of digestive enzymes, tukagira antibodies, hormones, discharge of neurotransmitter from presynaptic neurons. Ni ukuvuga ngo uburyo ibintu byose biba generated by a cell cyangwa se uburyo biba given by a cell, ibyo ngibyo byose tubibara muri exocytosis. Hanyuma rero this is the end of the chapter. Dukoze recording cyangwa se tugize recap tugasubiramo gatoya, nuko buri cell iri surrounded by a plasma membrane. Iyo plasma membrane ikaba ari cell semi-permeable. Hanyuma the presence of concentration and whatever itera movement of substances. Noneho plasma membrane mediates transport of smaller molecules by passive and active transport. Aho passive transport, ions ziva muri higher concentration to lower concentration nta energy dukoresheje. Hanyuma harimo nka simple diffusion, aho habaho movement of higher to smaller concentration, akaba ari movement ya solute. Hanyuma muri osmosis yo ikaba ari movement ya solvent. Hanyuma tukagira active transport, ariho concentration ikoreshwa iva muri lower concentration ijya kuri higher concentration, ariko tukanakoresha ATP cyangwa se energy. Then tugira icyo twita endocytosis, aho endocytosis ari uburyo twinjiza ibintu bifite biri large molecule. Hanyuma tukagira na exocytosis, ibintu aho bisohoka ari large molecules bigasohoka outside the cell. Thank you so much for your time. Thank you so much for finishing this video. If you have any question, just reach out to me in the DM. I will be more than happy to help. I think you'll find this lesson so helpful. Thank you so much. May God bless you.